Today we will discuss about macrophage lipid metabolism interplay with the pancreatic cancer cells. So in the previous video we have understood that how this classification of PDAC and tumor cells microenvironment tissues differ from the healthy patients that does not have the pancreatic cancer cells, and we have find out that endothelial cells are lower in the case of tumor populations compared to the normal cells, and the epithelial cells are more in the tumor populations compared to the normal cells. And now we will discuss whether this epithelial cells also, the macrophages amounts are also different in the case of normal cells and the tumor cells. So in this video we will find out whether there is a difference or there is a some with the pancreatic cancer cells and the normal cells. From the literatures, they have find out that macrophages macrophages has a role towards lipid metabolism. So in this uh, from this hypothesis, they identify the top 20 genes and its relevant score towards lipid metabolism. And these are the relevant scores of the top 20 genes which are relevant to the lipid metabolism. And among these, they are able to see that the genes that are in the second number is also the gene markers for the macrophages. This indicates that macrophages might play a role towards these lipid metabolisms, and they validated these markers in this. They validated their markers again in the Disney plot to confirm that these markers are present in the macrophages, and these markers are shown here, and most of their markers are resembles to the macrophages and also to the other cell populations categories. There we can see that the APOE markers also shown in the macrophages: TNF, C1QC, CD14, C1QA, and C1QB. And after that, they need to check whether there is a this macrophages plays an interrole or plays a role towards differentiation of the immune cells. For this, they did this monarch plot, and in this case, they can see the concentration of these markers for the macrophages at locations 1 and 3, and this interjunctions indicates the role of the cells towards the differentiations or towards this intercellular interactions. So this further substantiate their hypothesis that macrophages might play a role towards the intercellular signaling or differentiation of the cells. And after this, they check their some they check their they check the role of the macrophages towards differentiation into the different cell populations, or towards not a differentiation into different cell populations, but role of macrophages towards helping the other cells into differentiation. So in the normal case, they can see that the macrophages are more prone to differentiate towards its acinar cells, whereas in the tumor macrophages, tumor monocytes, whereas in the tumor, the macrophages are more prone to differentiate towards, more prone to helps in the differentiation uh, to form these epithelial cells. That's the reason epithelial cells are more in the tumor cells that we have seen in the previous videos. And after that, they decided to check what are the genes that are upregulated and downregulated in the case of these normal epithelial cells compared to the tumor epithelial cells. And they can see the genes that are involved in this genes that are upregulated in this uh, tumor macrophages, or genes that are upregulated in the epithelial cells are related towards this ER stress, and genes that are downregulated are related towards this apoptosis. And this is the core role of whenever the cells are under ER stress. And they validated whether and they also checked these features belongs to the which cluster of the cells, and they can see that different clusters, the same genes belongs to the different identity, and after that they want to check this after validating that these different features, these features not only belongs to a simple single cell cluster, but however it belongs to different cell clusters. They need to check what are the gene populations that are what are the kind type of cells that are downregulated or dysregulated in the control cells compared to the this pancreatic cancer cells. In this case, they have find out that M1 kind of macrophages are downregulated. M1 macrophages are downregulated in the pancreatic cancer cells compared to the control cells, where we can see that control cells has this M1 macrophage, whereas pancreatic cancer cells does not have those M1 macrophages. They are very low in amount. And after validating that it's the M1 macrophages that are downregulated, they need to check the M1 macrophages phenotypes in different cell populations, and from the statistical analysis, they can find out that in the case of normal cells, they can see the M1 macrophages are present to some extent in all the three normal cells, whereas when they check for the pancreatic cancer patients tissue, they can see very low features. We can see very low presence of a very low or negligible presence of the M1 macrophages that can be seen here, and they also quantified their this analysis, where they can clearly see that in the case of these acinar cells are significantly higher in the normal cells compared to the tumor cells, and same is the case with alpha cell beta, and the other structures are also shown that however for this M2 macrophages, they can see that increase in the amount of M2 macrophages is increase in the amount of M2 macrophages in the tumor cells compared to the normal cells. However, for the M1 macrophages, it's reverse, that M1 macrophages are more high in the normal cells compared to the tumor cells. So from this, they made a point that it's the M2 macrophages that might help in the lipid metabolism of the pancreatic cancer cells. And literature also shows that M1 macrophages has pro-tumorigenic properties, whereas M2 macrophages has anti-tumorigenic properties. So this so far their results, whatever they have got from the bioinformatics, is in line with the literatures, where M1 macrophages has pro-tumorigenic properties. So, and what they have find out is that M1 macrophages might help in the lipid metabolism of the pancreatic cancer cells so far till this experiment. So we will discuss further experiments in our subsequent videos. And if you need to understand more about their bioinformatic analysis in detail, such as what are the criteria they used towards this volcano plot, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.